Let's kick it off. So it is noon on the dot here. We are really excited to have our third and final um, presentations today for the Basin Breakthrough Seminar Series, featuring we've got Lower Basin researchers representing Arkansas, Louisiana, and Mississippi. And I will have MP, our moderator for today, do our official introductions of our speakers. But before we jump in, I just want to give a plug for our Early Career Confluence workshop that will take place at the end of January in Fayetteville, Arkansas. You can apply. I will drop the link in the chat in a second, but you can apply on the Sera-46 website. There is both the regular registration and the flight scholarship. We are covering all registration fees and meals from kickoff dinner to Thursday breakfast, and any materials and resources. And then we'll cover all the hotel accommodations for the first 20 people who will apply. And then we have 5 flight scholarships available. This should be a great opportunity for people to present their research, contribute to basin-wide research agenda for the Mississippi River Basin, as well as participate in some professional development opportunities. So if you have questions, just shoot me an email. I'll also drop that in the chat in a second. And then, yeah, you can apply online. And then with that, just some real quick ground rules before MP takes over. But each speaker will give a 15-minute presentation, about 2 to 3 minutes for questions. To ask your questions, please enter them in the Q&A chat box. During the talks is preferred because that lets us jump right into the questions. Or right after is also fine. We'll obviously answer as many live possible. And then if we don't get to your question, the speakers will have a chance to respond via the chat once we move on to the next presentation. And with that, I will go ahead and stop my share and let MP take over the duties here and introduce our first speaker.
Cool. Thanks so much, Shannon, and thank you for hosting this awesome and impactful series for the students up and down the basin. So today I'm going to introduce our first speaker. Kathleen Cutting is a recent master's graduate from the University of Arkansas in the Spears Lab, where she studied the impacts of intermittency and land use on agricultural stream nutrients and sediment export. She's continuing her journey in the Spear Lab as a program associate, leading a project on suburban expansion and stormwater resiliency, while assisting with other lab projects and conducting her lab analysis. Kathleen is especially interested in watershed scale research in agricultural areas and understanding how local agricultural conservation practices impact long-term watershed water quality. Today, she'll be speaking about how surface connectivity and land management drive divergent patterns in nutrient loss in a non-perennial agricultural stream. So Kathleen, with that, I'll let you take over and share your screen.
Yes, thank you, MP. I will share my screen now. Okay, so yes, hello everybody. My name is Kathleen Cutting, and I'm going to be presenting a little bit of my master's thesis work. I'm going to hold on and hide this floating meeting controls. But yes, I'm going to be focusing today on surface connectivity and land management and how that drives divergent patterns in nutrient loss in a non-perennial agricultural stream in Arkansas. Okay. So to begin, I'd like to give a few acknowledgements. So our funding sources from the Department of Energy and a University of Arkansas grant, as well as our nutrient analysis being run by the University of New Hampshire's Water Quality Lab, my committee members for their contribution and support throughout my master's work. Of course, current and former Spear Lab members shown in the picture. Big shout out to Shannon Spear for her mentorship. And then as well as the collaborators on the Quest project, which this research is a part of, which is studying intermittent streams throughout the United States. Okay, so to begin, non-perennial streams are globally ubiquitous, with over 50% of global stream length representing non-perennial streams. And that extent is expected to increase with climate change, which changes the magnitude and timing of nutrient delivery to our downstream systems. But non-perennial streams are not often protected or considered in water quality management and policy. And so therefore, it's important that Oh, well, non-perennial streams are often degraded through habitat, water quality, and functional integrity, making it important to study. Non-perennial streams are also critical ecotones at the terrestrial and aquatic interface, having an outsized influence on downstream water quality. And many of these overlooked systems are in agricultural landscapes. For example, in Arkansas, there's a large amount of poultry farming and beef cattle production. And from poultry litter, as a product from our poultry farming, poultry litter is applied to our pasture lands that then contributes to nutrient export downstream. And hydrologic alterations within these agricultural systems can ask and exacerbate intermittency through high irrigation use or compaction of our pasture lands. So this alters our nutrient transport in these stream systems from being transformers of nutrients to transporters. And this impacts downstream water quality systems such as Beaver Lake Reservoir, the key drinking water resource here in Northwest Arkansas. So the gap that we are trying to fill with this study is how intermittency and agricultural land use interact to alter downstream nutrient transport. And the objective of this study was to understand the temporal, so changes in surface connectivity, and spatial or land management controls on nitrogen and phosphorus dynamics in Brush Creek, Arkansas. So here we have Brush Creek, Arkansas, which is located near Goshen, Arkansas, Northwest Arkansas. And it is about a 5,000 hectare watershed and is a third order tributary to Beaver Lake Reservoir, that key drinking water resource that I previously mentioned. And Brush Creek is made up of about 46% forest, shown by the green in our figure, 43% pasture hay, shown by the yellow, and 10% other, such as unpaved roads, shown by the red. And there are up to 76 poultry houses in Brush Creek, shown by the black rectangles in our watershed figure. Okay, so to begin as well, it's important to mention that Brush Creek is primarily intermittent, many intermittent main stems and ephemeral tributaries. So in our figure, we have our study sites represented from upstream to downstream on our y-axis and sampling date chronologically on our x. And we show sites that are flowing through the blue color, sites that were pooled in yellow, and sites that were dry in the orangey red. And first, taking a look at our lower watershed, we see that it's primarily hydrologically active, which we represent to be surface flow, is what we were able to measure. And then in the middle of our watershed, we see our sites were primarily dried or pooled. Whereas moving to our upper watershed, we see that there's high surface connectivity in the middle of our water year, which indicates that our active period, or our period of high surface flow, is from January to May. And then our non-active period, or no surface flow primarily, is from October through December and June through September. So we took our samples in Brush Creek monthly at 20 different sites in the 2024 water year and sampled at base flow for nitrate and soluble reactive phosphorus. And at each site, we measured discharge using the cross-sectional area method, as you can see in the picture. Okay, so our first research question is, do changes in surface connectivity influence nitrogen and phosphorus export in Brush Creek? And we expected that nutrient loss would be greater and more uniform across the watershed during the wet season as a result of increased surface connectivity. And as we can see, under low connectivity, we expect more patchiness, whereas in high connectivity, there's that higher uniformity of nutrient export. So to begin answering our first research question, we used the metric of leverage, which is the influence of a subwatershed on an outlet's load. And negative leverage values indicate enrichment or solute production, zero values meaning minimal processing, and positive values indicating dilution or solute removal. And if we take a look at our figure, we have nitrate leverage on the top and SRP leverage on the bottom, with high connectivity shown in blue and low connectivity shown in brown. And we see that under high connectivity, there are differential patterns in nitrate and SRP. First, looking at nitrate, we see that the upstream is contributing to nitrogen loss at the outlet, so enrichment of nitrate. Whereas with SRP, we see it being more of a neutral transporter, so there's limited processing of SRP. Whereas if we look at our low connectivity period, we see that it's more similar through solute removal of derived from our upstream sources, likely with nitrogen being through denitrification and uptake or sediment absorption for soluble reactive phosphorus. And then we looked at another metric of spatial stability, which is how consistent areas of a watershed maintain their relative pollution levels over time. And this allows us to predict how one sampling can predict long-term pollution patterns. So high spatial stability, meaning that we would sample less, vice versa with low stability. And spatial stability values range from 0 to 1, with values closer to 1 indicating higher spatial stability. And if we look at our table, we have high connectivity periods and low connectivity periods for nitrate and SRP. We see that nitrate is more stable across the year and across these periods, meaning that the drivers of nitrate loss are more consistent regardless of connectivity. Whereas SRP is less stable at high connectivity, which was surprising to us, as there's variability in site characteristics through landscape or sediment dynamics. And then we looked at our active and non-active periods at the upstream with, again, active periods, so surface flow connectivity shown in January through May, and our non-active periods in October through December and June through September. And in our figure, we have outlet nitrate yields and outlet SRP yields in the active and non-active periods. And we see that during our active periods in the upper watershed, meaning increased surface connectivity, that the upper watershed contributed to higher outlet yield for both nitrate and SRP. Whereas if we move to the relationship of percent upstream sites flowing and our nutrient yields, we see that nitrate yield increased at the outlet. Whereas with SRP, we don't see that relationship significantly, but there could be a potential threshold effect with more of a relationship with increased percent sites flowing. And finally, our second research question is how land management impacts the patterns of nitrate and SRP export in Brush Creek, expecting that both nitrate and SRP loss will be greater in subwatersheds with more animal agriculture, so that cattle and poultry in our watershed, as runoff from animal production serves as a source of nutrients to adjacent streams. And here we have our relationship with our varying land use categories. And we see our main stem sites represented by our dark blue points and our tributary sites shown by our light blue points, with nitrate yield on the top and SRP yield at the bottom. And today we're going to focus in on nitrate yields, as this is where we showed our significant relationships. And so we have, as the percent of pastured hay increase, our nitrate yield increased. Similarly, as our active pasture land, which we measured through 2 windshield surveys, seeing if there were cows active or present on our pasture lands. And so as that increased, our nitrate yield also increased. And then as our percent forest increased, non-surprisingly, we saw nitrate yields decreasing. But if we focus in, we see that our tributary sites had higher nitrate yield, as they typically have closer connections to local areas with more connection to the landscape there. It is important to mention that we did not find a relationship with the number of poultry houses in Brush Creek watershed, as that was another key landscape influence in this watershed. So, in conclusion, spatial variation in both nitrate and SRP loss is more uniform at low connectivity across the watershed. So, there's consistent solute removal and higher spatial stability. Where at high connectivity, there are higher loads of our nutrients, which may be a silver lining to increase intermittency. Not to say that increasing intermittency is a good thing, but it allows us to have less nutrient export at these times, and we can predict how nutrients move downstream more easily. In addition, pasture and hay is a likely key contributor to nitrate loss from our watershed, not SRP. But it is important to mention that our study was at base flow, so we may have missed key transport times of SRP during storm flow, which can be explored in other studies. So in all, in agricultural watersheds, increasing intermittency and periods of low connectivity may decrease nutrient loading downstream and increase the predictability of nutrient patterns across our watershed. Emphasized by our site here with on the left showing a dry period and reconnection on the right. And with that, thank you for your time. And are there any questions?
Thank you so much, Kathleen. That was an awesome presentation. I really enjoy students when they, post pictures in the field and during the
Yes.
presentation because you get to see the hands-on connectivity of the project, plus all the analysis that goes into it. While we're waiting, so it looks like,
Sure.
Sorry.
Would you like me to unshare my screen?
Oh, I mean, either way, in case questions come up, I did have one. I'll start the questions
Sure.
off while we're waiting on people to post in the chat. Did y'all see any like seasonal trends associated to your nutrient studies?
Yes, that's a good question. So, in our wet season, so that kind of periods where we see higher connectivity that occurs in the spring and the winter, we do see higher loads of nutrients there. Whereas in the summer, we see lower connectivity, so periods of more pooling where denitrification often happens and higher SRP concentrations were occurring as it's released from our sediments in those pools.
And on one of the last few slides, you showed that decreasing trend in the forested areas. It's kind of interesting because down here in South Louisiana, we typically use those as like a wetland buffering zone or a forested area as nature-based solutions. And so, do you see that as an opportunity for some of these areas that have high loads of nutrients coming in these areas?
Yes, for sure. Vegetated riparian areas are a great idea to reduce nutrient runoff, as well as vegetated ditches from roadside management perspective can help reduce not only sediment transport but nutrient transport as well. And so in the future, looking at riparian area is a good predictor of how that can impact nutrient loss. Okay, then maybe.
Give it a few more seconds to see if any late questions come through the chat. And then if you do have any questions that might pop up, Kathleen, if you don't mind staying on and checking that chat as they come through and responding to those.
Yes, of course.
Awesome.
Yes.
Well, what we'll do is we'll go ahead and transition to our second speaker. So Kathleen, I'll let you stop sharing for a moment. And Animal, if you start getting ready, I'll do your introduction here. So our next speaker today is Animal Money, who's a PhD student in the School for Plant, Environment, and Soil Science at Louisiana State University. He also completed his master's in the same department where he developed a soil test-based sulfur fertilizer recommendation for soybean production in Louisiana. His current research focuses on the spectroscopic characterization of fluorescent dissolved organic matter in environmental systems with an emphasis on understanding its dynamic and implications in water quality. Today, Animal will be speaking on to us about the spectroscopic characterization of fluorescent dissolved organic matter and agricultural runoff. So with that, I'll let Enamul share his screen and start his presentation.
Hello, everyone. Can you hear me? And is my screen okay? Okay, thank you. Hi, everyone. Good afternoon. My name is Enamul Hakmoni. I'm a first year PhD student at the School of Plant, Environmental, and Soil Sciences. Um, I'd like to thank my professor, my advisor, MP Hayes, for the opportunity he gave me to work in his lab. As he said, I was working with nutrient management for 2 years in my master's, and now kind of shifted a little with the nutrient movement and changes within the agricultural systems. So today I'm going to be presenting my first projects and the preliminary data that I've got so far. This is entitled as the understanding of the retention of nutrient using fluorescence dissolved organic matter in the diverse soil substrate mixture. So I'll present today is the natural and dissolved organic matter and why this is important in the agricultural system and few techniques that I have been using in my lab to characterize the dissolved organic matter. The details about the experiment that we have completed so far and the future projects that we'll continue to work on. So what is basically natural organic matter is? It is the fundamental component of the soil and the environmental system, actually. And this is very complex and can be available in different forms. It also sorry. It also has a crucial role in the environment too. So it can act as buffer solution in the a buffer system in the soil solution for the nutrient uptake of the plants. It can also supply nutrients as a source for the crop production, like nitrogen, phosphorus, sulfur. One of the most important water quality characteristics is the light penetration into the water. The normal natural organic matter also controls that. The most crucial thing that I'm interested in is the influence of the movement, binding, and bioavailability of the nutrient that is also affected by the dissolved organic matter or the natural organic matter. Uh, so we can divide the natural organic matter in many ways. For example, based on the source material, the sources that the natural organic matter that has been derived from terrestrial source like plants, leaves, etc. They are called allochthonous organic matter, and the organic matter that has been produced within the system. For example, in aquatic system, algae is developed within the system that is called autochthonous organic matter. Based on the molecular weight, we have higher molecular weight and stable organic matter that is refractory and often labile and lower molecular weight that is supposed to change within the system, that is labile organic matter. We can also classify them based on group, humic and non-humic groups. Um, so the non-humic in the non-humic groups, they are carbohydrate, protein, and fats, and in the humic groups, they don't have any single formula yet. So this is very crucial. We should develop a system or a technique to characterize those dissolved organic matter because these are the most reactive part of the soil organic soil solution, which reacts with nutrient and interact with plants in the nutrient uptake. So on the left side, we can see a Venn diagram that showed the proportion of the different types of organic matter in the um, ecosystem in the environmental system. So the purple circle here, that is our main concern, the dissolved organic matter, and which is separated from other type of organic matter by a fine filter that is often used in the analytical lab. As in, if I'm recall, the measurement is 0.45 micrometer, but sometimes some scientists also use 0.2 micrometer or 0.7 micrometer, but most often it is defined by 0.44 micrometer. Uh, you can also see here the yellow circle. So the dissolved organic matter is also associated with the nutrient. Here is dissolved organic nitrogen, DON, or phosphorus, DOP. On the right side, here's the diagram that shows how the agricultural system carries out the dissolved organic matter, uh, nutrients, pollutions in the surface water with runoff. So the rain events, the irrigation into the crop fields, um, and the waste that is generated in the livestock and poultry also carried out into the surface water and added nutrients and creates impairment of the water. Now, the the way I'm talking, like I have mentioned so many times the dissolved organic matter, but now we will only consider a portion of that, a little portion of that, which is optically active. That means if we excite excite the organic matter with light, it can absorb the light, and also it can emit the light in a longer wavelength that we can catch in the ultraviolet and visible spectrum region. Those signals are very crucial and very unique characteristics for the sample that we are talking about. So these signals that you can see on the right side of the graph here, this is an excitation emission 3D plot. This is very unique chemical in a unique structure for a specific chemical, which can be changed sample to sample based on their molecular compounds and functionality, based on their source and history. So this actually essentially gives us a few background information about the source, about the structure of the dissolved organic matter that we are talking about here. So we can use this technique further to characterize the DOM in any system. So one of the ways we analyze our samples in FDOM analysis is the ultraviolet and visible spectrum absorbance spectra. So for example, the absorbance at 254, it gives us an idea about the aromaticity of the carbon, often leads us to the labile fraction of the DOM. The A350, which represents the refractory part of the carbon, which is a high molecular weight and more stable than the labile portion of the organic matter. The S275 is essentially absorbance slope, which is inversely proportional to the molecular weight of the organic matter. That means if we have higher S275 value, the system has lower molecular weight of the organic matter. And if we have normalized all these absorbance with total organic carbon, which we'll do in future, if very soon we hope we will get total TOC analyzer, so we'll do that. If we normalized these absorbance parameter with total organic carbon, we will get the SUVA ratio, which is also a great parameter in the AFDOM research. Now, another way we analyze our samples is the excitation emission matrix. We have, when we run any sample, we have a wide range of excitation wavelength and a wide range of emission wavelength. But scientists and researchers find out few chemical indicators which are very crucial in identifying the characteristics of the organic matter involved in the sample. For example, fluorescence index gives us idea about the source of the organic matter that has been in the system. It tells us is the organic matter comes from terrestrial source or aquatic source. The biological index, for example, is also known as BICS. It represents the freshness of the organic matter present in the system. Lower Biggs value means there is very little recently produced organic matter there. Same goes for humidification index. The higher humidification index means the organic matter present in the system is more complex and more humic in nature. Another strong statistical tool that we're planning to use is the parallel factor analysis or parafac analysis. Um, what happened is when we analyze the sample and get the excitation emission metrics, it actually gave us the history or type of origin of the organic matter presence in the system. But if we coupled the parallel factor analysis with the excitation emission metrics, it gives us the idea of which component is there, and we can separate the components based on mathematical term. Now we have seen the use of Webdom so far in water treatment ecosystem research and marine environment, but so far there is very little work has been done in the agricultural system. Um, so we have written and submitted a review paper, and we have started working with the use of Abram in the agricultural system. So our next experiment, my first project would be also use of Abram in the agricultural system. So this is an experiment where we have been using 8 different treatments. Or is first one is the control peatlight, second one is 10% biochar amended peatlight. Third is 20% biochar amended peatlet on the top, and the lower end is only the control peatlet soil. Uh, the fourth one is the control peatlet soil over stratified over the 20% biochar amended peatlet. Um, so we have bark certification, we have peatlet soil certification over here, and in the last treatment, I want to emphasize on that is it has uh, 20% biochar amended bark under the control peatlet soil. So we have a very specific result for this treatment. We'll see in later slides. Our main objectives here to observe the effect of soil substrate and nutrient retention and how the FTM characterization could be useful in these analyses. So we're using samples from a greenhouse treatment, which is very alike with nursery operations in the United States, and which is very important because a nursery contribution uh, contributes about $400 billion in the US economy and 200,000 jobs in the green economy. It created by nursery industry. So we have collected the samples from them, and as I say, like we calculated the chemical indicators as I discussed earlier, is 275. We have seen the general lower or decreasing trend uh, within a 6 weeks period, uh, which essentially means that overall gradual flushing of higher molecular weight, as it is inversely proportional to the molecular weight of the organic matter. If we can, we see closely, we can see the more clear trend here. When the biochar is at the bottom layer, we can see a drastic decline of the S275 value, which essentially means that the biochar acts as a strong absorbents in the system. So initially, we can infer that the biochar or the architecture of the substrate in the pot has a great impact on the retention of the different types of molecular weight of organic matter. If you talk about humidification index or Higgs, we also see the same type of trend here. In terms of treatment 8, where the biochar was amended with bark and placed under the control peatite soil, we see that a drastic decline of humidification index after 1 month or 4 weeks period. Although overall the Higgs value is here higher, that means a high humidification is there, but it gets lowered here after weeks of 4. It also represents that uh, humidification index also here represents that we have more complex and higher molecular weight of organic matter present in the sample. Now, if we look into the biological index of Biggs, it indicates the freshness of the component. But overall, it has a very low range of value right here within all the six periods. And we can clearly see that the Biggs value is lower. That means there is very little amount of recently produced organic matter present in there. It also proves our type of treatment we have used, like biochar and bark. They're terrestrial substance, and they need more time to degrade and produce a uh, biological, recently organic matter. And in terms of treatments, we see the similar trend where the biochar is in the lower layer. It shows different than other treatments of the experiment. Uh, Now, the fluorescence index also confirms the value confirms that we are using the terrestrial components for our treatments. So to conclude, we can say that the biochar layer actually acts significantly in the retention or absorption of the organic matter, and also the results are consistent throughout the all the chemical indicators we have calculated so far. Um, and after analysis can actually explain the temporal DOM stratification. The one message I would like to share from this preliminary data I have so far is that substrate architecture has an impact on the fate of the DOM and the transformation. So we're planning to continue our work and we'll run paraffin analysis. We're planning to have a TOC analyzer. We'll run that. And one of my lab mates, she runs gallery instrument to have the idea about the nutrient. We'll try to collaborate with that too. Um, and so this is all from my presentation. If you have any question, please, the floor is yours.
So we just have one moment for questions before we need to go into our next speaker. I will say if you do have questions and we're not able to get to them on this live feed, then please send them to the Q&A link below, and Emma will be answering those during the next presentation. Looking to see if any are coming through right now. I'll pose one quick one for you. You know, currently, are there any kind of reference points, or can we reference SUVA between different agricultural systems? Because I know this is a nursery-based, project. How might this link to other, research in the basin like Kathleen just presented on?
So from a literature, I haven't seen that much work within the agricultural system. Um, so that's why we're working on it. So I think the base after getting the baseline data, we'll be able to uh, say that. And if we can use the TOC analyzer, we can connect different systems together with total organic carbon, and we can say that later.
Cool. Well, Animal, please be looking for questions in the Q&A chat down there. And if you don't mind stopping sharing, I'll be introducing our third speaker for today. So Lane Galloway received a bachelor's degree in crop and soil science and a master's degree focused on nutrient management from Auburn University. Currently, he works at Mississippi Water Resource Institute as a research associate and graduate student. The research he works on involves monitoring water runoff, effects from changes in crop planting or fertilizer management on small plot research and on-farm experiments. The main goals with this research are to reduce environmental footprint of crop production practices and improve the economic productivity for growers. Today, Lane will be speaking to us about the implementation of site-specific management and alternative crop systems to accelerate nutrient load reduction. Lane, thank you so much for joining us today, and I'll turn it over to you if you want to start sharing your screen.
Hello, thank y'all. Thank you again. Hello, my name is Lane Galloway. I'm a graduate student with the Mississippi Water Resources Research Institute. I'm looking forward to talking with y'all today. Uh, maybe, um, so yeah, I'm gonna look into some of our research that we have going on and the implementation of site-specific management and alternative cropping systems to accelerate nutrient load reduction. So to start off with, agriculture is a major cause of non-point source pollution. This is a map showing the total nitrogen and total phosphorus, the percent of total nitrogen, total phosphorus, eventually making its way to the Gulf from the highlighted areas, which includes all of the Mississippi River and its associated tributaries. And I just want to quickly mention that uh, being part of the lower Mississippi River Basin, we have the Mississippi Delta in the western part of the state, and 90 to up to 100% of the total nitrogen, total phosphorus, that if it is reaching a stream or water body in this region, it is eventually making its way down to the Gulf. I mean, we do have a large impact on the Gulf and maintaining its water quality. I want to quickly mention a few things about our common corn and cotton systems within the Mid-South. Typically, our row spacings are either 96 or 76 centimeter row spacings, 38 or 30-inch row spacings. We typically grow on raised beds, and often what we do is with conventional tillage, disking in the fall, and then using some form of a hipper or roller hipper to make those beds for those rows. And also, majority of the time, our fields are uniformly managed. So, and what I mean by that is the inputs that we use, like fertilizer or seeding rate and other things, is uniform for the whole field. So the rate is the same across the whole field. And we do not think that we are optimally applying our inputs on our fields because simply fields are not uniform. It's common to have yield maps like this where you have low yielding zones, which here are shown in the red, or high yielding zones shown in the green. And uniform application of inputs like nitrogen or phosphorus can limit our growers' net returns and productivity. Now, a lot of the times, the most fields they'll have this variation due to just simply inherent variation in the field that's not easily fixable. Some of these things could include soil texture or soil drainage, as well as elevation. All of these things could not simply be changed by uh, management for the whole field. And so we want to look into more site-specific management on these fields, and doing this through variable rate application of those inputs. The main goals from the research that we conduct is determining the relationship between nitrogen rate, nitrogen transport, and runoff for our common nitrogen fertilizers. And we do this through some rainfall simulation experiments. We are also examining some variable rate application of inputs like nitrogen to see how that affects grower net returns, and then the effects to nitrogen loads and runoff from the changes implementing from the variable rate application of those inputs. And then we also want to look into determining if we can integrate some alternative management components into a system to improve grower productivity and water quality. And things like this include alternating row spacing and tillage practices. So to start off, I'm going to talk about some of our rainfall simulation experiments. We have done this to mimic a large rainfall event immediately after a nitrogen application. And so we want to observe if nitrogen source and rates affect nitrogen loads within, or how they affect nitrogen loads within surface runoff. We conduct these in Brooksville, Mississippi, on one of Mississippi State's agricultural research stations. The rainfall simulators that we use We have constructed them based off of methods laid out in paper, Humphrey et al. And I want to point out just a few things from that. So it our simulators have a one nozzle at the top that is designed to mimic natural rainfall, and we calibrate these to put out 70 millimeters or 2.8 inches of water per hour, which would be considered a large rainfall event. We are doing this on a 3 square meter plot. All of the runoff is then collected on the downslope end in a trough that is then funneled into a bucket where then we are pumping the water into a tank that we can agitate to get water samples. We have done this experiment with a urea rate experiment, and a urea ammonium nitrate or UAN rate experiment where we're analyzing runoff in runoff amount of volume, water pH, temperature, and nitrogen analysis. All of our water samples are then analyzed by an EPA certified laboratory. So quickly, I want to look at some of our results from UAN. So we did this in 23 and 2025. Our urea ammonium nitrate application rate here is on the x-axis, kilograms of nitrogen per hectare, with total nitrogen loss and runoff on the y-axis. And we observed that with an increase in our UAN rate, we saw a linear increase in total nitrogen loss and runoff, where we observed that as 1 kilogram of nitrogen, 1 kilogram of nitrogen being applied, we saw about 10% being lost in runoff from applying UAN. Now looking at urea, with the same, we saw urea application rate here is on the x-axis, total nitrogen loss and runoff on the y-axis, where as our rate increased, we saw a linear increase in total nitrogen and runoff. Here with urea, we observed a 2%. So as 1 kilogram of nitrogen being applied, we saw 2% of that being lost in our total nitrogen and runoff. However, these are small scale rainfall simulation experiments, and I mentioned before, most fields are not uniform, so we want to scale up this research and monitor water runoff on a whole field scale. So, and we are doing this also looking at variable rate application through site-specific management. And the goal here is to optimize the distribution of inputs like nitrogen and phosphorus and to improve grower net returns. Most growers have some form of variable rate application technology, or they have equipment that can be set up with this technology. A lot of growers already do have uh, uh, a lot of data on yield, soil sample data, or just overall just a lot of data that simply are just not using. Also, there are a lot of growers that are using variable rate application for some of their inputs, maybe like lime or one input or the other, but not a whole lot of growers are using variable rate application for all of their inputs or on all of their fields or for their whole system. But there's a lot of reasons why variable rate application hasn't been used by a majority of growers. What we're going to do is we're going to focus on is some on-farm research locations throughout Mississippi where we are focusing on extreme field variability. So this is one of our fields where this is yield map here on the left, about a 200-acre field, and we're then setting it up into two management zones. So we have this low yielding zone here shown in the red, which was about 55 acres. So we're setting up two zones, more of the higher yielding or average yielding area here in the green, and the lower yielding zone shown here in the red. And this low yielding zone here, about 55 acres, was due to a lower cation exchange capacity compared to the rest of the field. And this was due to a different soil texture in that zone, and this is not something that could simply be changed with management, or it's just an inherent variation in that field. And so we set up these uh, replicated blocks within that zone where we're altering the nitrogen and seeding rate. Also on these, we're mostly focusing on corn production systems, corn and cotton production systems. So to look at this one field, this is some of our preliminary data. This is the nitrogen here was applied in 3 applications. So we implemented 2 seeding rates and then 5 nitrogen rates, and we implemented a variable rate application for the side dress, which was done a few weeks after planting. So some of the nitrogen was applied as urea at planting, and then a UAN application for our side dress, which is what we did our variable rate for, and then some urea also being applied around tassel. So quickly, I want to look at the net returns from this field. So with the uniform rate and the higher seeding rate compared to our lower seeding rate and lower nitrogen rate, we saw that in both, we saw a negative net returns. However, with this lower seeding rate and lower nitrogen rate, we were able to increase the net or increase the net returns by $15,000 for that low yielding zone. And for the whole field, we increased the grower net returns by $192 per hectare. And by applying less nitrogen in that low yielding zone, we decreased the nitrogen being applied by 10%. And the goal here is to show that these low yielding zones, we can apply less nitrogen, reduce any losses off of that field, or increase grower net returns on that field, and at the same time reducing the nitrogen that could potentially be running off of these fields. So now we are in the process of evaluating water quality on these fields on a whole field scale. We're doing this through implementing water runoff equipment from Teledyne ISCO. We have some fields on farm locations where we're setting these up on the edge of those fields, and where to catch all the water, or where we can hydrologically isolate one field compare it to another field where we implement uniform changes, and then one side will have variable rate application. And then we are also implementing some of this research on some of Mississippi State's agricultural research stations. And this one here is located in Brooksville, Mississippi. And like I mentioned, we're looking at alternating management components into a whole alternative management system. So we have some previous research showing that a narrow row spacing can increase yields, implementing some minimal tillage. Also, we had a graduate student that was looking at some nitrogen application, and we saw that with UAN, which is usually applied in injected into the soil into a trench, by closing those trenches, we can reduce the amount of runoff coming from those fields. And this is an experiment that is going to be a long-term scale in Brooksville, Mississippi. And the main thing I want to point out here is each of our plots are hydrologically isolated, so that we can monitor all the water coming off of each of these plots. We'll quickly mention we're looking into different crop production systems, looking at some of the common production systems in the Delta and the prairie region of Mississippi, as well as some of the conservation systems composed from the NRCS. And then what we are planning to implement or propose is an alternative management system with narrow row spacing on wider beds, implementing minimum tillage. And so from our rainfall simulation experiments, we observed roughly about 10% nitrogen being lost with UAN and 2% being lost with urea through water runoff. We know variable rate application of fertilizer can improve grower net returns. And also we evaluating runoff water quality from these fields to observe the effects from implementing those changes and how it also can improve water quality for our region. Want to mention that our research is sponsored by the Mississippi Corn Promotion Board as well as the US EPA. I went through that very fast, but if anybody has any questions, I'd be happy to take them. Thank you.
Thank you so much, Lane. Really appreciate the presentation. It's really cool when you can get some of these isolated field trials going on where you can have the individual components at these experimental stations. It does look like you've got a question there. The question, I don't know if you can see it in your little Q&A box, I was wondering your thoughts about how this can be scaled up. What kind of data is required across the fields to efficiently implement target nitrogen application to low yield areas?
Yeah, so that we are in the process of scaling this up. So we have some on-farm research trials where we are implementing some variable rate application. Now, we are implementing these where into these replicated blocks. Now, we are not changing the whole field into management zones. The idea there is to implement all management zones. So, like you have a low yielding zone compared to higher yielding zone, but we're not changing the whole field. There'd be a lot of risk associated to the grower for doing that. So we want to show that it is something that it can be done to potentially increase their net returns. And on the water quality side, we are setting up these systems to monitor water quality. And here being the start of November, about to move into the rainy season, so hopefully we'll start getting a lot of water samples on our fields and start collecting that data and see what we find. But these are ongoing. Hopefully that answers your question.
Thank you, Bree. Great question there. I had one more for you. Have y'all done any comparative study, looked at other universities that might be doing this with similar crop types for a comparison of what y'all are doing?
I don't know. I don't know of anyone else that is. I know there is some other researchers doing some variable rate studies, and also monitoring water quality. I don't know of any, as of right now, I don't know of anyone else that is specifically doing or starting to work on implementing this and also monitor the water quality at the same time.
I dropped you a link, a direct message to the Purdue Sin Lab because they do some really cool work with tile drainage. In this area, it sounded like it might be comparable systems, maybe a little bit different, but worth checking it out because there's some great collaborations across the agricultural networks up and down the basin.
Yeah.
With that, I'm most likely going to turn it back over to Shane for some closing remarks. Thank you to all the speakers today. Great presentations, y'all.
Yes, thank you everyone for participating. Excited to put a close on this first seminar series, and hopefully we'll have more coming up in the future. And just a final plug, please apply for our workshop. It'll be super fun and all that, and you can do that on the Sera-46.org webpage under events. So we hope to see you there. All right, thanks everyone.